What do I always say? Hi, this is Miss Lynn. Welcome to my period. Wonderful. Wonderful, fantastic period 3 class. I actually have something to say, and I'm glad. I'm going to tell you wonderful, and I'm going to try to do it without crying this time. Try. I wanted to tell you, and this is just an introduction. Sorry if you're not here, but my absentee children banding away. This is for you too. I wanted to tell you here at the start that I am thankful for you. That I chose to be an educator. I had considered going into medicine. That was my thing all along. And then I just thought, I'll try teaching 1 year. And then here I am 31 years later. And I wanted to tell you how thankful for you that I am. Because I know sometimes you are exhausted, you are hungry, you are tired, you are overwhelmed, you stay up late finishing an assignment. Maybe you're doing membeans or making boards. What were the boards you were making? Yes, that. Doing math practice problems, college board practice problems, and your little eyes want to close, but you stay up late and finish, and you come to class and you're hungry and beat down, but you still generally always walk in here with smiles on your faces, ready to learn, which makes it easier for me when I'm trying to teach. And I appreciate all of the hard work, and I know each and every one of you is really trying to do the best they can and navigate through your first AP class. And I wanted to tell you because at my Thanksgiving table, we go around and talk about what we're thankful for, before we eat. And I wanted you to know that I'm going to say that I'm thankful for you. And I'm thankful that I get to work in a school like this, to get to work with students like this, that I can trust you in my room. You're polite. And a lot of teachers don't get to have that. And I just wanted you to know from the bottom of my heart that I appreciate all of the hard work that you're doing. And I know you're struggling to do your best, and sometimes it works out for you and sometimes it doesn't. But I hope that at the end of this year that we are together, that you have learned some study strategies that you can apply to some of your other courses, and that you feel good about what you've done. And know that when I push you and ask you to do this or that I'm trying to do that for your best interest, because I want you to be the best you that you can be. So, in this Thanksgiving season, I just wanted to say thank you. All right. Sorry. Okay. All right. So, I'm going to back up a little bit, and no mascara today. I'm going to back up just a little bit so we make sure that everybody is on the right page with our thinking with chapter 10. So, first thing I want to make sure we address. And what you see me addressing right now is because I've just gone through period 1, and I know what questions they asked, so I'm assuming you might have some of those same questions. So the first thing we need to remember is that all of your body cells, for us, the type of organism that we are, all of our body cells are diploid cells, except for our gametes. Those are the only cells in our body that are haploid. And when you form, if you have a female body, okay, you do something called oogenesis. And when you do oogenesis, you are going to make eggs. And I'll talk a little more details about that in a minute. But what you're trying to do is generate a haploid cell, one that has half of each homologous pair. Now, who formed your homologous pairs? You have, how many chromosomes do we have total in our body, by the way? 46, okay? 23 of those came from your biological mother, and 23 came from your biological father, yeah? So you have 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s, etc. Now, you have 22 technically homologous pairs, and in our binary world, if you are a female in this room, you really have 23 homologous pairs. Because the 23rd pair are the sex chromosomes. And if you are a female, your 23rd pair is what? XX. So that is a homologous pair. Now, the 23rd pair for you males in the room is X what? Y. Okay? So that is not homologous. What is on an X and what is on a Y are totally two different things. And we'll address how that plays out. But when you do oogenesis, ladies, you've got to generate an egg that does not have 46 chromosomes in it. You want it to have how many? 23. Yeah, you want it to have 23 chromosomes. And that is referred to as your N number, is your haploid number. So I could say haploid or N, right? And then the sperm that's arriving at the scene, do you see, and this is not even drawn to scale, but inside of that sperm, okay, also would have 23 chromosomes, N, right? So when that sperm fertilizes that egg, the 23 chromosomes it has, okay, are brought in. Now 23 and 23 are what? 46, and that equals our what number? 2N number, 46, okay? That is our 2N number, our diploid number. So I can describe this as either N equals 23 or 2N equals 46. I am saying the same thing either way, right? So potatoes, their diploid number is 48. Their diploid number is 48. So identify with your bio buddy what is their 2N and what is their N number. Their diploid number is 48. Right? So when I say their diploid, di means what? 2. That's their 2N number, right? And so their diploid number, their 2N number would be 48. Their haploid number, their N number would be what? 24. So when potatoes are having potato porn, okay, when they are having sex, right? So when they are fertilizing, when that pollen is fertilizing that egg, okay, they're each going to have 24 chromosomes, which will join together to form what? 48. Yes? Haploid, diploid. Ask your bio buddy, are you on solid ground, haploid, diploid? Okay, then oldest bio buddy, could you please talk about this right here? Go ahead, oldest bio buddy. The naked family with the giant naked baby. Oh no, the baby has a diaper on. Okay, no. Yeah.
Thank you.
Basically, you want to develop and they each produce eggs and just like combine together.
All right.
Wait, that's so good.
Okay, so mom and dad, right? Diploid bodies. In meiosis is happening in the male gonads, the testes. The only thing that are haploid are not his testes, but the actual gametes, right, are haploid. And same thing for the female. They get together through fertilization. Obviously, they did not draw all the chromosomes. It would just be hard for you to see the total 46 chromosomes in their diploid zygote. You got that, right? Okay, so when we talk about homologous pairs, remember they are the same size, the same shape, and they code for the same what? Traits and characteristics, right? And our next chapter, we're going to talk about Mendel and how that plays out in our phenotype and the way that we look. But during the S stage of interphase, what's going to happen? Yeah, DNA replication is going to take place. And at that time, when DNA replication takes place during the S stage, you're getting ready. If you are a spermatocyte getting ready to form sperm, you're getting ready to do meiosis. So what's going to happen is you're going to be in S, and then what stage follows S? G2. Then you're going to do meiosis 1, interkinesis, and then you're going to do meiosis 2. That's the way it's going to play out. Okay? And there are different things that can happen, but basically in meiosis 1, to summarize what happens in meiosis 1, is you have got to separate the homologous pairs from each other in meiosis 1. Meiosis 1 occurs in just one cell. Just one cell doing meiosis 1. When you're done with meiosis 1, you will not have one cell, you will have what? 2 cells, because that's what happened when a cell divides. Okay? Each of those 2 cells are going to do meiosis 2. Okay, so you'll do meiosis 2 in 2 cells that you just formed from meiosis 1, and there you will separate the sister chromatid. All right. Now, key events can happen throughout each of those that increase variety. Why is variety important? Because variety, that's what you need. That's the precursor for natural selection, right? Because natural selection is assuming that some traits are better than other traits. And sometimes environments change. And so you want some different traits maybe than what you had before. You can get a genetic recombination through sexual reproduction. And we want to identify those places where it happens. So the first thing is, we're going to go through prophase 1, metaphase 1, anaphase 1, right? We're going to go through those. But some key events will occur. In prophase 1, the chromosomes become what? Visible. And we still have what else happens? The nuclear envelope and the nucleolus disappear, radiating microtubules, asters, synapses. Okay? In synapses, it's not that you're going to have an exchange of sister chromatids, because all that would do, it would be like these, exchanging white flower for white flower. Is that going to get you any variation? No. What you're trying to do is exchange white flower with one of the other non-sister chromatids for purple flower, whatever this is, right? And when they do that exchange, that's going to increase variety. Whose turn is it, young or old? Yeah. Young. I would like you to explain, please, crossing over. All right. So let's talk about this crossing over. At that place where it does cross over, what we're looking at right here, chiasmata. Say that. Chiasmata. Okay, so you're exchanging. Now, the process of the homologous pairs coming together, synapsis, forms, you could call it a tetrad, a bivalent. These are all names that work. These are homologs that have come together. Now, when does that occur? During prophase 1. And the crossing over occurs during prophase 1. This is really good because this ensures, if you do synapsis, chromosome 1 from mom's got to hook up with chromosome 1 from dad, and 7 with 7, and 8 with 8, and 9 with 9. So we get all the right homologous pairs together. Yeah? That's critical. Then once we've done some crossing over, which can happen multiple times along that length of those chromosomes, or no times, but it could happen multiple times. Now, none of these chromatids are exactly the same. And since every one of these chromatids is different, that means every single gamete formed is going to be what? Different. And this is where we're looking at only one homologous pairs. One homologous pair. How many homologous pairs do we have? 23. So can you see how we can have so much variety in the sperm or egg? Because this is just looking, this is a different, right? All 4 of these chromatids are different as a result of crossing over. And so you will have 4 different gametes for sure, regardless of any other chromosomes, because that is different. But we have 23 homologous pairs all doing crossing over. So that's one area. Okay, ask your bio buddy, are you square with crossing over? Do you have it? Do you have questions you want to ask?
So basically,
whenever you have something like where you're doing this, tell me, okay? Because I want to make sure we totally and absolutely understand this without any problems, okay? So ask that question. Any questions so far? Okay, so following crossing over in prophase 1, what stage is going to follow prophase 1? Metaphase 1. They meet in the middle at metaphase 1. Did I teach you the song? No. Oh, we're going to learn the song. Okay, let's learn the song right now. Okay. I wish you could see me, but you can't. Okay. We might record this, and then we can put it on YouTube separate. Oh, wait. We can. I'll just put it on YouTube. You don't have to film you if you don't want to see it. You can just film me. I'll teach it to them. Okay, wait. I just need my regular video. Ooh, I'm not smart. Video. Are we good? Do you need to back up? Am I too close to you? You might need to stand up. Okay. Just stressing me out. Okay. So tell me when. Okay. So this is a song for those of you that are absent from class today. I'm going to teach everyone the song for my meiosis, and we're getting ready for what? Sex. Okay, so show me your 2 chromosomes. Show me your 2 chromosomes. Now, there's no cell that only has 2 chromosomes, but we just don't have enough fingers to do this otherwise, okay? So how many chromosomes are in this cell? 2. So the diploid number is what? 2. The 2n number is 2, right? We're trying to make cells that have just 1. That would be not the diploid number, but the haploid number, the n equals 1. What happens to these chromosomes during the S stage of interphase? They replicate, okay? These are still homologous pairs. They were this was a homologous pair before. Here's a homologous pair after. Yes? Okay? So when we do this, this is going to occur during the S stage, but we can't see them yet. When do they become visible? Prophase. So the first division we're going to do is meiosis 1. And the first stage in meiosis 1 is prophase 1. So we're going to go like this. The chromosomes become visible. You already know this part. The nuclear envelope and the nucleolus disappear, radiating microtubules, asters, and then go synapsis. Okay? And then during synapsis, what can occur? Crossing over. Perfect. Then you're going to go like this. Crossing over has occurred. We'd have to chop off our fingers, but we're not going to do that. Then the next stage is they meet in the middle at metaphase 1. Who's meeting in the middle? Homologous pairs. Good. Away at anaphase 1. Who's going away? Homologous pairs. 2 nuclei at telophase 1. Cytokinesis. Interkinesis. This is in between, okay? Now each of these cells generated are ready to do meiosis 2, which is going to occur in 2 cells. These 2 cells right here. We still have the sisters, right? But because we separated the homologous pairs, each of these cells is considered haploid. Why? Because they don't have their dance partner anymore. Mommy, daddy divorced, right? They've been separated. Does that make sense? Is it helping? Okay, so now we're going to do prophase 2. They're just visible. Here we go. Oh, sorry. Do you need, wait, now they'll have to switch. Go back, because otherwise I'll do it closer. So these are in 2 different cells. So prophase 2, metaphase 2, just like before. What do you think is going to happen next? Anaphase 2, telophase 2, cytokinesis if we had a hatchet, okay? Then we will make 1, 2, 3, 4 cells. Each one of them will have how many chromosomes in them? 1. Ah, okay, perfect. Okay. Oh yeah, I don't have to do anything with it. I did, for TikTok, I did it in 2 stages. Do you want to try to do it just in one fell swoop? Yeah. In 60 seconds? Okay. Does anybody want to stand up here with me? You want to practice once? Yeah, okay. I can't see any of it. Okay. When we practice once, I'm going to try to look at the clock so we can do it, and I'm not going to add in all the descriptors, okay? But we'll try to do it. I'll try to do it how I would do it. Okay, we're going to do meiosis, getting ready for sex. Okay, show me how many chromosomes you have. 2. How many homologous pairs? 1. Okay, what happens during the S stage? Good. Okay, so what's the variant? We're going to do meiosis 1, and the first stage is prophase 1. Ready? The chromosomes become visible, nuclear envelope, nucleolus disappear, radiating microtubules, aster, synapsis, crossing over, then they meet in the middle at metaphase 1, away at anaphase 1, 2 nuclei at telophase 1, cytokinesis. Now we're ready for meiosis 2. It's going to happen in 2 cells. So here's prophase 2, metaphase 2, anaphase 2, separating the sister chromatids, and telophase 2, cytokinesis, making 1, 2, 3, 4 cells, and all of them are what? Haploid. Perfect. We can do that.
Wait, you didn't do interkinesis.
Oh, crap. Okay. Yeah. Cytokinesis, interkinesis. Remind me of that. If you start doing it, I'll do it. Anybody want to come up here and be with me?
Only Ricky. Ricky wants to go up, but he's too shy.
Yeah, Ricky is there.
We can all just do it. Is it going to be on back?
Okay, make sure you get the fingers. Oh, wait.
Ricky, just go up.
Is that better? Okay, are you gonna be ready? Wait, I gotta look at the clock. I need somebody standing behind her when she's starting to run out of time. I need you to like 5, 4, 3, 2, 1 me so we're not cut off. Well, and you're gonna nod when you hit play. All right, we're going to do meiosis now, both parts, meiosis 1 and meiosis 2. Show me your chromosomes. How many chromosomes do we have? 2. How many homologous pairs? 1. What happens during the S stage of interphase? And these are called what? Sister chromatids. This is still a homologous pair, right? We're going to start with meiosis 1. What's the first stage? Prophase 1. Here we go. The chromosomes become visible, nuclear envelope, nucleolus disappear, radiating microtubules, asters, synapses, crossing over, then they meet in the middle at metaphase 1, away at anaphase 1, 2 nuclei at telophase 1, cytokinesis, interkinesis, we're ready for meiosis 2, chromosomes are visible, meet in the middle at metaphase 2, away at anaphase 2, 2 nuclei at telophase 2, and cytokinesis. 4 cells, they're all haploid.
You got the end of haploid cut off.
Crap, let's go again. We were so close, so we have to go faster. So close. Okay, we're going to go faster. Yeah, what was I doing here? I don't even know. What was I doing? I went like this. I was stressing out. Like, how many seconds do we have?
Can I go up with you?
Yes, get up here. Anybody else want to come on? Ricky, get up here, baby. Yeah!
Wait, it has to be 4.
It is 4. Wait, I need people to say. Yeah! Okay, so let me see.
Chris, I'm going next to you.
I'll be in the middle, and then you can kind of pan around and see all of them, but stay close to somebody. You're going to get a minimum 5,000 views. Everyone. Can I have people fill in these spots, please? Fill in the spots. Oh yeah, sit right. Somebody can go like this. Do it right here.
Should we all just? I mean, someone who knows it because they
You see it? Turn around.
Miss, yeah, I took off this. I got 12 likes.
What?
I got 12 likes.
You got 12 likes? Yeah. Right here. What?
I got
Wait, can you see? Get up a little higher. I don't know if you see it. Okay, we're gonna do.
Go off like
Exit.
There you go. So I can see what she's doing.
Yeah.
Wait. Oh yeah.
All right, we gotta be ready this time.
Okay, ready.
3, 2, 1.
Okay, we are gonna do meiosis for you, both meiosis 1 and meiosis 2. Show me your chromosomes. How many do we have? 2. How many homologous pairs? 1. What happens during the S stage of interphase?
DNA.
And these are called what? Sister chromatids. So meiosis 1, what's the first stage? Prophase 1. The chromosomes become visible, the nuclear envelope and the nucleolus disappear, radiating microtubules, asters, synapses, crossing over. They meet in the middle at metaphase 1, away at anaphase 1, 2 nuclei at telophase 1, cytokinesis, interkinesis. Now we're ready for meiosis 2. Prophase 2, the chromosomes become visible. Then they meet in the middle at metaphase, away at anaphase, 2 nuclei at telophase, cytokinesis, and you have 4 haploid cells. Yay!
I was doing good at the. Oh, that's right. They're still recording.
Oh, I totally forgot. What should I do?
No, leave it.
Just leave it. Sorry. Oh my gosh. Sorry. I'm so sorry. We were trying to help you. Oh my gosh. There's like 5 minutes of nothingness. I'll put right here. Come back. All right. Pathetic. Okay. All right. So you learned the song. So now what I want you to do is you're each going to take a page, okay? And not it. Decide who's going to go first, okay? Break down what you see. Who's going to go first? You're going to take on this whole page. Describe what happens in this page, and then I'll see if you have any questions.
And there's 2 different types of variations.
And then the same thing crossing over. Except within the cells.
That's where we stop. I wanted to label it. Okay, sorry, band. Okay, so questions on this. What questions do we have? What do we see happening right here? Crossing over. Good. And is there evidence of crossing over here in metaphase 1? Yes, perfect. How many chromosomes are in this cell? 4. So what is the 2n number? 4. 1, 2, 3, 4. 2n number is 4, right? This is a diploid cell. We're going to make a haploid cell that has what? 2 chromosomes. It'll have 1 little chromosome and 1 big chromosome. Okay? So right now in metaphase 1, the homologous pairs have met in the middle. On anaphase 1, they are going away. We're separating the homologous pairs. Questions on this? Ask your bio buddy. You got any questions on this part? Oh, okay. All right, switch roles. Here's your next picture. Go. Yes. Wait, I'm going to answer it out loud when everybody's done. Okay, come back to me. So the nuclear envelope is reforming. Now, what might happen here is these chromosomes might decondense, unwind, decondense during interkinesis, or they might not. Okay. Cytokinesis, ladies in the room, in our binary world, you will not finish meiosis. You will stop. You won't do a complete cytokinesis because we finish that final cytokinesis if an egg gets fertilized by a sperm. Then we finish it up at the second mitotic division. So it depends. There's variations in how you go about this. Yes, you got to.
Does crossing over occur in prophase 1 or metaphase 1?
Prophase 1.
Okay.
Yeah, all your crossing over will occur here in prophase 1. By the time you get to metaphase 1, they're just all lined up here in the middle. Oh, and here's something I want to talk to you. In this case, the little blue lined up on this side, but he could have just as easily lined up on the other side, right? Do you remember what that is called? Independent assortment. So each homologous pair lines up in the independent independent of the other. It doesn't matter as long as the pair's in the middle. It's not like everybody goes, hey, dad's on the left, mom's on the right. It just lines up independent of each other. Okay, any other questions on this? Yes?
What's the difference between cytokinesis and interkinesis?
Oh, cytokinesis is division of the cytoplasm. Interkinesis is that time between meiosis 1 and meiosis 2. Okay, that time between meiosis 1 and meiosis 2. Okay, what else? These are all great questions. All right, switch again. Now you're doing you're going to do meiosis 2 in 2 cells. So here's one cell across the top and another cell across the bottom because it's happening in 2 cells. Discuss, and then I'll do questions. Go.
So you're just going to go through prophase 1.
So basically those get separated.
Okay, questions? Yes.
Interkinesis is just sitting there basically.
Yes. And interkinesis might be like, interkinesis done, and it might pass right through it, especially for you guys that are making sperm, right? Because in the time I say for you guys making sperm, you made like 1,000 sperm, okay? So you're just like, you blow right through interkinesis. You're like, and keep going, okay? But it depends on the organism, how much time they spend there. Okay, other questions on this slide right here? Feeling strong? Yes.
Wait, in prophase 2, are those, is it 2 homologous pairs?
No homologous pairs. And that's why at the point where you're here in prophase 2, look what it says, N equals 2. You only have 1 large chromosome. Its mate was over here, right?
Okay.
So you have separated the homologous pairs, and that is by definition what makes it haploid, because you do not have its matching homologue, right? They're not together. And so even though it consists of sister chromatids, and you would say to yourself, how can that be how? It's haploid because now you only technically have 2 chromosomes, remember? Because as long as the sister chromatids are together, they are considered 1 chromosome, as long as they are together. So it doesn't have its partner, no dance partner. Therefore, no crossing over, right?
So
No
those
crossing
are,
over.
So those are sister chromatids.
These are sister chromatids.
Okay.
Yeah, sister chromatids. So meiosis 1, we separated the homologous pairs. Meiosis 2, we separated the sister chromatids. Perfect. Okay, what else? More questions? All right. So go to the last slide. Here you go.
So basically the same thing. I guess it's separate.
Okay. So these could be, hey, there we go. These could be 4 sperm that you just generated. They are all haploid. They have how many chromosomes in each one of these? 2. So when a haploid 2 gets together with another haploid 2, it'll make a diploid 4. Or if a haploid 23 gets together with another haploid 23, it'll form a diploid. Yes. Questions? Let's have them. Yes.
So for this, is it the same thing? Like, is this specifically for like sperm or like? Is it
I will show you. Let me jump ahead. Show you something that's kind of interesting. Maybe. Oh my, I passed it. Huh. Here we go. All right, look at this. Here's spermatogenesis, the beginning of sperm. Okay, so here's that parent diploid cell, the spermatocyte, and meiosis 1 is occurring, and you're taking your 2N, you're separating your homologous pair, so each cell is going to be what? N, right? Now, but each one of those N chromosomes still consists of what? Sister chromatids. Then each one of them is now going to separate the sister chromatids for humans, right? That would be in this, right, there'd be 23 chromosomes, 23 chromosomes lined up right here, yes? And 23 chromosomes lined up right here. I'm not going to draw them all. And then you would separate that out, yeah? And you'd have 23 here. And you got that, right?
Well, I was thinking that there was only 2 because in all the pictures there's
Right, but that's because they're too hard to draw 46 chromosomes and all those cells. Yeah. Yeah. So then you will make 4 sperm that are all haploid. Okay? And that sperm can fertilize an egg. Now let's talk about females. We do oogenesis. Now, little history of the egg. The egg, during ovulation, pops out of the ovary and it moves into the oviduct. Another name for the oviduct is the fallopian tube. As it will take a few days for it to move through the oviduct, and then if fertilization is going to take place, it's going to occur in the oviduct. Side note, you could have sex on a Tuesday and fertilize your egg on a Thursday. And the reason is the sperm will remain in the female genital tract and they'll just be like, we are ready, okay? And they will just sit there and hover, you know. Half of them will go the wrong oviduct because they don't ask directions, but they will just be sitting there. That was a slam. Only you got it, too. Oh, you got it, too? All right. But what will happen is those sperm can be just waiting in your oviduct. Some sperm will last like 4 days. And, yeah, powerful sperm. But then the egg pops out, ovulation, and the sperm is like me, okay? And so then it'll fertilize. That's what I'm saying. You could have sex on a Tuesday and fertilize on a Thursday. But then that egg is, see, the egg, once it pops out, it's only viable maybe 12, you know, some window around 12-ish hours. And so that's when a sperm can fertilize the egg. But after that, if it doesn't get fertilized within 24 hours of ovulation, it's not going to happen. And so once it gets fertilized, it has to move down the remaining bits of the oviduct and down into the uterus, and then it's got to implant in the uterine lining. So that whole travel time, that egg is going to need nutrients. So it's the females who pack the lunch because the males are just trying to get there as fast as they possibly can. So they're very streamlined, very small, right? This is not to scale. This would be a ginormous giant of a sperm, okay? It would just look, you know, like that, okay? So they're bringing nothing to the picnic. We have to pack the entire picnic lunch. So when we do cytokinesis, we do it unevenly. We don't do it in the middle, cytokinesis. We do cytokinesis. And that way we have one giant, do you see right here? Here's one giant one that needs to undergo meiosis II and this little itty bitty polar body. You cannot fertilize that polar body. It doesn't have any of the proteins and the organelles that it needs. This is just going to deteriorate. It might undergo meiosis II. It might not undergo meiosis II. Okay, so here, this one, here's meiosis I has just taken place. Now, this one right here has to undergo meiosis II, right? And when it undergoes meiosis II, it's not going to bother until a sperm says, I'm here. And you're like, oh, you're serious. Okay. And then we will finish. So that's why your eggs that are sitting, they're sitting like this stage right here. They've done meiosis II, but they haven't done the final what? Cytokinesis. So right now, ladies, however old you are, that's how old your eggs are. So my eggs are 53 years old. These are some old eggs, right? 53 years old. Leave those alone, okay? They're old, okay? So that process is you make one large egg because you do uneven cytokinesis. Good? All right. So did I answer your question that you said? All right. Boom, 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 boom. We were here. Other questions about this? Okay, let's write the notes on it. Here we go. Meiosis I begins with one diploid cell. Begins with one diploid cell. Prophase I, this is a what step? Key step. Synapsis occurs, forming tetrads. You could say bivalents. And synapsis is occurring, joining the homologous pairing together, right? Putting them together, putting the homologs together. Crossing over occurs between homologous chromosomes in the tetrads, specifically the non-sister chromatids will do the crossing over. Crossing over increases genetic variation. Areas of crossing over form a chiasmata. The nuclear envelope disappears, you already know that, allowing the spindle to attach to the homolog. In metaphase, the homologous pairs line up in the middle. In anaphase I, what happens? Homologous, what? Homologous pairs separate. Yeah, way on anaphase. Homologous pairs separate. Telophase 1, same as before, 2 nuclei form, except it may not be followed by cytokinesis. Then you have interkinesis, which is the resting stage between meiosis 1 and meiosis 2. Meiosis 2 begins with 2 haploid cells generated from meiosis 1. Prophase 2 is same as before, except there's no crossing over. Why is there no crossing over in prophase 2? There's no homologs, no homologous pairs. There are no more homologous pairs. They have been separated. Metaphase 2, chromosomes line up in the middle. This time, single file, just like you do in mitosis. Single file. In anaphase 2, what's happening? Sister chromatids separate. Telophase 2, 2 nuclei form, followed by cytokinesis. Okay, let's watch a little video about this. Anticipate the stages that they're showing you. Okay?
Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug of war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gamete.
Okay, that's it. All right, ask your bio buddy. Did you understand that? Did you have any questions? Questions, concerns? Yes.
Okay, so regarding crossing over for
Yes.
sister chromatids, do they cross over? It doesn't do anything, or do they just
I don't
never
think
cross
they
over?
don't cross. I mean, is it possible? Sure, anything's possible. It just wouldn't change anything.
Okay.
Yeah. What else? Okay, let's test you out. I want you to log in today as something that you are proud of yourself about that you accomplished this week. You're like, that's right, I did this. That's what I want you to log in as.
a song that helped make
Ooh, fun. Yay! Not crying every day. Oh my gosh. Okay, so put them together. And those of you out there, I will do it with you. Look, watch me, watch me. I'm putting prophase 1 here. Now, if I put the wrong one, look what happens. Okay? So, prophase 1. What one do you think matches prophase 1? All right, let me put it in there. I'll try it. They're doing it on their own, so look through those and tell me which one you think I should put. Yes. Which one should I put? Tell me. That's right, this one. Is anybody done? Okay, what should I put for telophase 1? 2 haploid cells? All right, what follows telophase 1? Prophase 2, what should be prophase 2? In 2 cells. Metaphase 2? Chromosomes in the middle, good. Yay, boogie down. Okay, I got another one for you. First on top, last on the bottom. When you do it, you get it right, come up to the board and drag them in the right order up here too. Because the people at home need to see if they're still watching, because I forgot to turn off the video when we did our TikTok. TikTok foul. Yeah, come on up, drag them in order. Keep it going.
Wait, do we just go again or should we let it?
Good job.
Can I do another one? Hold on, this is fine.
Keep going.
I'm too short for that.
Yay! All right, let's move on. Comparing and contrasting mitosis with meiosis. So here, these pictures are in your book too, so you can use them as a study guide. So here's meiosis. You can see the 2 divisions, and then here is mitosis. There is a chart where meiosis 1 is compared with mitosis, and where meiosis 2 is compared with mitosis. So when you look at these, if I say sister chromatids are separating, that is true, right, of mitosis in anaphase. So that would just say anaphase. If I was referencing mitosis, it would just say anaphase, where in meiosis it would say anaphase 2, right? Is that also true of anaphase 1? No. It's not. But if you look at meiosis 2, anaphase 2 and straight up anaphase, which just means if it doesn't have a 1 or a 2 behind it, then it's referring to mitosis, the same thing happens. Sister chromatids separate, right? Look at the whole thing. Is it exactly the same? Yeah, it's the same description. So if I ever ask you a question on a quiz, like a map question, multiple answers possible, and I say sister chromatids separate, you could say what? Anaphase 2 and anaphase, straight up anaphase, right? Check with your bio buddy and see if they understand why. And tell them why you wouldn't say anaphase 1. Okay, you can't say anaphase 1 because what do you separate in anaphase 1? Homologous pairs. That's why you can't say anaphase 1. There's also another chart that just straight up contrasts meiosis with mitosis. And look at your notes right now where it says meiosis compared to mitosis. Occurrence. Very common for mitosis, all tissues for growth and repair. Meiosis only at certain times in sexually reproducing organisms. Number of divisions. For mitosis, how many divisions do we have? 1. Whereas in meiosis, we have 2. In anaphase 1, we separate what? Homologous pairs. In anaphase 2, we separate sister chromatids. On number of daughter cells, how many daughter cells do we have for mitosis? 2. How many for meiosis? 4. Results for mitosis are diploid, identical cells. For meiosis, haploid and not identical due to crossing over an independent assortment. Now, I have something I want you to add in the mitosis column underneath results. You can do mitosis and go from diploid to diploid. That's the only thing we do mitosis for. But other organisms, not us, but other organisms, remember how we looked at plants and the haploid generation and alternation of generations, remember the gametophyte? They will do mitosis, they will go end to end. They can go 2N to 2N and also end to end. You're keeping the chromosome number the same. If you're keeping the chromosome number the same, whether it starts with a diploid cell or it starts with a haploid cell, that is mitosis. So I would add underneath results, diploid, identical cells, 2N, arrow to 2N, or you could put in parentheses, N to N. Check with your bio buddy, make sure they understand that.
Do you understand that?
And I will refresh your memory in just a moment if you forgot, okay? Not it. Just decide if you're going first or second. You don't even know what you're going to do. Just decide first or second. Okay, so what I want you to do is take Mr. Froggy life cycle, if you're going first, and identify on the arrows. Do you need to identify it with mitosis, or do you need to identify what's on the arrow with meiosis? Decide first with your bio buddy.
Oh, you're smart. Alright, so you've got. Okay, so. A tad from a tad.
Okay, I'm jumping in. When the tadpole becomes a tadpole with legs, do we do mitosis or meiosis? All right. When we go from tadpole with legs to young frog, what are we doing? Mitosis. Young frog to adult frog? Mitosis. Adult frog to eggs? Meiosis. Egg mass to tadpole? Mitosis. Got that? Okay, whoever was in charge of that life cycle, hush hush, other bio buddy, take on the fish. Did you just switch seats or something? Oh, I just heard like, I'm like, it's not enough.
growth and then half.
Whoa. Oh, it's like seven. My
Okay, so any questions on that? Pretty straightforward? Yes.
So is like fertilization like kind of its own stage? Or?
It's an event. I wouldn't say it's a stage, it's an event where the two nuclei fuse together.
Okay.
So if, like in humans, if you fertilize an egg, it doesn't mean you're going to be pregnant. You're not pregnant until that fertilized egg actually implants in the uterus. Because fertilization occurs in the oviduct, implantation occurs in the uterus. So there's a difference between those. I don't want you to just like, oh, it means, yeah. All right? Okay, now, remember this, alternation of generations. I'm just using it with Fern. Okay, I would like the youngest bio buddy to go first with the diploid stages and the oldest bio buddy do haploid and then switch. Start each time at fertilization. Okay, but you each get to do a half, you know, just take turns. Go. Okay, I'm jumping in. Did you get to go? Do you need any more time? I'm sorry. Okay, here's our zygote. Okay, here's our zygote. And from this zygote, who is diploid, because sperm, egg, right, diploid zygote, it's doing mitosis to form an adult fern plant. You know fern plants, some of you might have some in your house. Okay, peek on the underside of the fern leaves and you might see some sporangia. It's like orange little dots. So you remember last time I taught you sporophytes, right? Do you remember that? Say it with me. Sporophytes always make spores by meiosis, right? Say that again. Sporophytes always make spores by meiosis. Gametophytes always make gametes by mitosis. Put it together. Sporophytes always make spores by meiosis. Gametophytes always make gametes by mitosis. Okay. So here in this sporangia, they're going to make these spores by doing what process? Meiosis. What are they getting ready for? Sex. You always are getting ready for sex when you do meiosis. So, but they start off by making these spores that are haploid. These haploid spores do mitosis and become this gametophyte structure. Here is the gametophyte right here. It's like a heart-shaped structure. And that's what it is for fern. And it would be like small. You could see it with your eyes, but it's very, very small. Here, and this is in a moist area, because right here, different color. Right here, okay, is where you have the archegonia, right at the top of this heart, at this point of the heart right here. You have the archegonia, which is making the female egg by mitosis because it's already haploid, right? Okay, and then down here, usually there's like, kind of looks like hairs kind of coming off here. That's the antheridium, and there the sperm is got to swim over to the egg and fertilize it. And then you have a diploid zygote right here, and the sporophyte, because ferns are seedless plants, okay, they didn't evolve yet to have seeds, that's the sporophyte grows right up out of the gametophyte and becomes the dominant plant, right? It's not like any of us go, look, I have a fern for you, and it's this little bit of gametophyte. No, we have the ferns in our house or in our gardens or our yards are all the diploid sporophyte that grew up out of the gametophyte. Okay. On your notes, go to where it says life cycle and plants, alternating haploid and diploid stages. The sporophyte is usually the dominant plant structure, and it produces spores by what process? Meiosis, that develop into the gametophyte. And the gametophyte is a haploid structure that produces haploid gametes by mitosis. Right? Do you see the irony there, right? You're making gametes, but what are you doing? mitosis. Why are you doing mitosis? Because the structure that's actually making the gametes is already haploid. So since it's already haploid, it's not going to do meiosis again and make them like a quarterploid or something. And then we saw this already. We discussed this. It's in mini-me because I don't think they're going to ask you about this at all. But you can see spermatogenesis. The location is the testes. It results in 4 haploid sperm. Oogenesis results in 1 egg. In B, cytokinesis does not occur until the sperm penetrates the egg. And cytokinesis is done unevenly to keep the nutrients in 1 egg cell. Okay, let's discuss fails. Okay, when it does not work out. Okay, so you can have a chromosomal mutation. It can be a change in the number of chromosomes you have, so you have too many or too few. Maybe you have a whole extra set of chromosomes. Okay, we would die if that happens. Or it can be a change in the structure of that chromosome. Those are the kinds of chromosomal mutations you have. So, non-disjunction is failure to separate. Now, you could fail to separate in meiosis 1, or you could fail to separate in meiosis 2. Oldest BioBuddy, you're going to take this slide on. What is going on? Is it going on with the egg? Is it going on with the sperm? Did it happen in meiosis 1? Did it happen in meiosis 2? Go. Okay, is this an egg problem or a sperm problem? Egg problem. And you can see it happen during meiosis 1. So the homologous pairs, instead of going away at anaphase, they said stay at anaphase. And the problem with this is all of the eggs are screwed up. Because half of them have too much and half of them have too little. And that's why you have half with 2n plus 1, 2n plus 1, and the other half 2n minus 1, 2n minus 1. Okay? Contrast that with what's going on here. Other BioBuddy, go.
It's not the homologous.
What failed to separate here? Sister chromatids. Is this as bad as the first non-disjunction we saw? No. Why isn't it as bad? Yeah, these eggs over here are normal. Just these half are screwed up. Does that make sense? All right. Did I talk to you about amniocentesis?
Oh, yeah.
A little bit? Remember when somebody's pregnant and they can take a big needle and remove some of the fluid? Okay. What they do is they stain the chromosomes, and it is a karyotype. A karyotype is a photograph of the chromosomes. So they catch some of the baby cells that have been sloughed off into the amniotic fluid, and they find one that's going through mitosis. They stain the chromosomes, and a computer will put them together via their homologous pairs. So look right here. Here is a karyotype. Is this a male or a female? Male. So you can see all the homologous pairs 1 through 22, and then here is 23 right here, and we can see an X and a Y. All right? All the first 22 for male are all homologous. Now, it's going to blur a little bit, okay? But each member of the homolog consists of sister chromatids. Can you kind of make them out a little bit? Okay, held together by a centromere right there. If we look up here, maybe you can kind of see. Can you see the two? Okay, so meiosis 1, if you were going to do, these cells are not going to undergo meiosis. These are just cells that are skin cells or something that have come off of the baby. So they're only going to do mitosis. They're just lining them up here so you can see the pairing that you can do. Okay, look at this. Boy-girl issue, what do you see? Okay? So, is it a boy or a girl? It's a boy because I can see the XY right here. But here, this right here, the chromosomes 1 through 22 are called autosomes. Your 23rd pairs, this 23rd pair, your XY or XX is called your sex chromosomes. Trisomy 21, which is what this is the most common autosomal failure where you get non-disjunction. So as a result, they have 3 chromosomes right here. That's never good for us, never. As humans, plants, they can have an extra one of every single one of these. They're called polyploids. They usually have really big fruit, and most of our wheats, varieties of wheat and strawberries are polyploids because it makes the strawberries so huge. So, but that doesn't work with us. Plants are different. It doesn't work with us. So trisomy 21 is the most common autosomal failure where you have non-disjunction. I don't want to say the Down, the Down syndrome is not a failure. I'm just saying failure to separate. They are sweet and wonderful and loving if you know a Down syndrome child. Adult. Oh, here's the question for you. You're going to tell me, it's me, I made the mistake. All right. All right. But you at least realized that it was a mistake. Okay, do you remember the story about Goldilocks? Some of the porridge was hot, some was too cold, and some was just right. Okay? So just right porridge, okay, is called euploidy. Maybe you can think you are eukaryotic, euploidy, okay? And that is just right. If you have a whole extra set, so that's way too much, that's called a polyploidy when you have an extra set, okay? And then if it's just incorrect, aneuploidy. And against, opposite, I guess you could think of it like that, so it did not work out. All right, now let's take a look again at those sex chromosomes. So a female is XX, a male is XY. Can you see the discrepancy in size there? Now, think about this. If I've told you it's bad to get an extra chromosome, think about this. If you have 2 X's, do you see how much extra stuff you have? Clearly, males do just fine, and they only have 1 X. So do you see how that could maybe be a bad thing if we had 2 X's? Okay. Now, we do them. But what happens in our cells is one of our X's condenses into a bar body. Now, a bar body, remember when we studied euchromatin and heterochromatin, DNA that's active and not active? And non-active DNA winds itself up tightly and is called heterochromatin. DNA that is active and being used at that time is euchromatin. So those of you that are female, in every single cell in your body, one of your X's is condensing. And you don't know which X will condense. Okay? And that is called a bar body. So you remember, one of your X's came from your mother and one came your father. So you're kind of a mosaic depending on what X is active. This is seen really clearly in calico cats. Do you know what calico cats look like? Kind of blotchy, different colors. That's what they look like. And the reason is because their color of their fur is triggered by which X chromosome they have that's active. So you do not have male calico cats in the sense they don't have the coloring like that. They'll just be solid, one color. But the female is because it depends on which X chromosome is active and it affects their coloring. Do males have bar bodies? Do males have bar bodies, the condensed X? No, because they only have 1 X. Now, there are syndromes where you are male, but you are XXY. That is called Klinefelters. So you have an extra X in there, but you're still a male because you have the Y chromosome, and the presence of the Y chromosome will trigger testosterone, so you will develop male. Will they have bar bodies?
Yes.
Yes, they will have bar bodies even though they are male. Okay, and in reality, this is a nucleus right here. This is the cytoplasm. This is the nucleus. Here's the nuclear envelope. You can see the bar body right there. And it's a condensed X chromosome. Okay? Not it.
Not it.
Explain this. Okay, so let's look at your notes. On non-disjunction, on 10.6, changes in chromosome structure, number and structure, a non-disjunction failure of either homologous pairs or sister chromatids to separate. A karyotype is a visual display of the chromosomes arranged by size, shape, and banding pattern. Euploidy is the correct number of chromosomes. Autosomes are homologous pairs 1 through 22. Sex chromosomes, 23rd pair of chromosomes, they are not homologous in males. Bar bodies, an additional X chromosome that condense and can be seen under the microscope, 1 in normal females, 0 in normal males. All right, try this. And if you're done, you can come up and drag it in here. Go right up there. Good job. What a team. Yay! All right. Here's another one for you. I just got like where I was like doing these. I must have just figured out how to do them or something. I got some new music on this one. When you're done, come on up. Yay! Good job. Okay, let's look at some examples here. Down syndrome is trisomy 21. They have identified what actually causes the issues with Down syndrome, the having trouble with some mental development, changes in the shape, in the tongue, in the digits of the fingers, all of the things that go along with that. It has to do with them having an extra copy of the GART gene. If somebody has trisomy 21, but they don't have an extra GART gene, they do not develop Down syndrome. So it's that gene in particular that gives them those characteristics. Here you can see this would be like a normal female, 2 X's. Now, even though one of the X's condenses, right, into a bar body, if you do not have 2 X's, which may have something to do in development, you will have issues. You usually have a barrel-chested, short in stature, can have some learning disabilities, can have heart problems, can have kidney problems. and actually can have webbing at the neck coming down here for Turner syndrome. I'm going to tell you, we have several Turner students in our school. So be very careful how you discuss it. Of course, you would never mock anyone, right? That would be inappropriate. But I just want you to show that sensitivity to that. Because you could see somebody, and I could be Turner's and you could just not even know it. I'm not Turner's. I would, of course, tell you if I was. But you have trouble with fertility. That's a real big deal because your ovaries will not mature. And as a result, you could possibly still have a baby, but not from one of your own eggs because your ovaries have trouble. And so to keep them developing along the female pathway, they usually have to give them hormones in order to do that. Okay? Klinefelter's, like I mentioned, is XXY, and there are issues associated with that as well. So on your notes, you have Down syndrome. It is the most common autosomal trisomy. You are short in stature, heart problems, intellectual disability. Chances of this syndrome increase with the age of the mother. Why? Her eggs are old and they do not do they fail to separate. They do non-disjunction, right? They fail to separate. Those are the autosomes. Changes in sex chromosomes, XO. O means you don't have a second chromosome. That would be Turner syndrome, female, short in stature. Usually lead normal lives with hormonal supplements. You're not going to have a bar body. Why? Because you only have what? One X. XXY is Klinefelter syndrome. You are male, increased risk for some disorders, sterility, long arms and legs, and you will have a bar body. There's something called triplo X or poly X females. They are not ba-ba-ba-boom. Okay? They actually are tall, slender. They tend to have wide set eyes, and there have been, like, when I can never find a picture of a poly X. Nobody has admitted to being a poly X, but they had models that they think that are poly X based on some of their characteristics, but I would never put that up because that would be not appropriate if, that's, anyway. And then Jacob syndrome is XYY. Do you see that right there? Now, XYY, there are proportionally, if you go into a prison and you do normal, chromosomes, euploidy to Jacob syndrome, the percentage is higher in prison than in the general population. Some people say that extra Y chromosome makes them more what? Angry and violent. Okay, but now I'm going to tell you something else. Children who are XYY, they can have some learning disabilities. They can have really bad acne. They can have trouble with talking. And then I am confident that I went to school with a boy, his name was Lars, who was XYY. Now, once I learned about the characteristics. And you know what? He was marginalized because he acted so just a little bit off. Terrible acne, terrible. Felt so bad for him because of that extra, testosterone. Because if you are struggling with acne as a male, or female actually, it is because of all the testosterone you have, okay? And what do sometimes people do to somebody who is different? Make fun of them, judge them, right? Marginalize them. So is it possible that it's not because of nature and their DNA that makes them more violent, but because they have been marginalized so long and bullied, that tends to make them violent, right? And then that's what ends up putting them in prison. Do you see what I'm saying? So we have to look at all the things. All right? Then the last two are pretty easy. Changes in structure. Basically, these have to do, yes?
How do you get an extra Y? Because if the mom has two X's and the dad has one Y, where's the Y?
Because remember sister chromatids and non-disjunction. The two copies of the Y fail to separate in meiosis 2. Does that make sense? Okay. Good question. So changes in chromosome structure, basically these are screw-ups in crossing over. So in the case of a deletion is, hey, I thought we were crossing over, and you send over part of your chromatid and nobody gives you anything back. Then the other one will have extra and you won't have enough. And then another failure is during crossing over, you flip one of your chromatids in the crossing over process. It doesn't line up right and it gets flipped. So your genes are in reverse in that one segment. Sometimes that causes no problems. Sometimes that causes big problems. And then the last one, translocation. Remember, you're supposed to do synapsis with your homologous pair. And if you don't do synapsis with your homologous pair, so if chromosome 2 hooks up with chromosome 7 and they cross over, they're not exchanging the same information. They could be exchanging foot structure for heart structure, right? Not good. So on your notes for that one, deletions are missing a portion. Duplication is you have an extra segment. Inversion is a flipped segment. All right, and let's do a practice quiz, and then you can go hook up with your team. You can hook up with your team as soon as you're done. You can move to a lab bench or a grouping here. And then here, I'm going to show you the questions so you can see them. It's question 1. Try and answer it at home. Question 2. Question 3. Question 4. Question 5. Pause it if you need to, okay? Because you should try to answer these to get ready for your quiz. Question 6. Question 7. And question 8. As soon as you're done, start looking at your highly suggested reading and thinking. Some of you need to move faster so we can provide feedback. I got some of you at 4 and some of you at 8. I'm going to pause just for a minute. I'm going to stop it so you can see your results. Here you go. I'll let you look through and see what the answers are. Okay, study, study, study. Sorry for that huge part in the middle where we forgot to pause it. My bad. Okay,